these singlet and triplet states. But it's very interesting physics that's going on in here. And going back to fluorescence, again, I think Professor Potma went through that. So the energy of the fluorescence is lower, of the fluorescence photon is lower than the energy of the absorption, and that really means that the emission, the fluorescence is redshifted. The shift between the is redshifted with respect to absorption. The shift is called the Stokes shift, and this really helps. So the larger this Stokes shift, the better you can separate emission from absorption. This is an example of you can use multiple fluorophores to label a cell in this case. So you can label nuclei, like mitochondria is labeled here in green, and then microtubules. So this is how you can play with multiple fluorophores, and I think many of you know that. You can have linear and nonlinear fluorescence. Again, Professor Potma went at length through the difference between these two types of interactions. One thing I wanted to make sure, or I just wanted to emphasize a few aspects here. So first, I wanted to make sure you understand the difference in the wavelength involved in these two processes. So in a single photon excitation, you have this type of, I would have liked a laser pointer here. This doesn't work well, but so you have this excitation with a single photon that's characterized by a certain energy, right? When you have two photons involved, the energy of the photons is half of that excitation. So if you use like a UV or a blue laser line to excite to have the one photon excitation, for the two photon you would use a laser with photons that have half that energy, and half that energy in terms of wavelength, because of this relationship, it translates to twice the wavelength. So usually typically for two photon fluorescence, two photon microscopy, we use near infrared laser. And another important aspect is the time duration of the pulses. We use pulse laser for two photon microscopy, and that's because again, going back to Professor Potma's lecture this morning, he was talking about the lower probability of the nonlinear processes. So to have this process happening over the one above, you really need to work with high peak power to have a lot of photons. And high peak power you created by using these short pulses. So usually you will use femtosecond pulses, 10 to the minus 15. Power is energy per pulse. If you deliver this bursts of pulses very short in time, you create high peak power and can make this process more likely to happen. I think that's all clear, right? Also, oh yeah.
For the single photon excitation, there's
Brenda, can you, for the sake of the recording?
Oh yeah. Can we use an IR for the single photon excitation too?
Most molecules have the absorption in the UV. You could design molecules to excite them longer, but probably most of them are in the UV. Most of
Brenda,
the molecules of interest.
Reconnect to the lecture I gave yesterday. I chatted about how when you use, typically, when you use wavelengths or energies in the blue to green to yellow, you're really exciting electronic energy states. But as you get to the red and near infrared, for most molecules, you only have enough energy to populate vibrational states. And fluorescence is predicated upon populating the first excited electronic state, which is called the singlet state. So if you can't get to the first singlet state of the molecule, you can't generate fluorescence. Okay. That's another way to address that question.
That's
but people are
correct,
engineering contrast agents that have singlet states that are lower energies, and you can populate using lower energy photons.
Okay, so yes, I wanted to summarize the origins of contrast we have available when it comes to fluorescence. So we can use labels such as free dyes and nanoparticles that may bind to a specific region of a cell or tissue, or conjugated to antibodies, receptor proteins, or other biomolecules of interest. If we are interested in probing calcium signals, would use indicator dyes that dynamically bind an ion and then change either fluorescence intensity or emission spectrum. Or we can use fluorescence proteins, such as green fluorescence protein, that are produced by organism after the DNA is altered to express that particular protein, so to track a particular or specific cells. Or we can use intrinsic fluorophores, and I'm going to talk about that a lot. So these intrinsic fluorophores, they occur naturally in the system under study. In our case, skin, where we use a lot endogenous fluorophores such as NADH, FAD, collagen, elastin, melanin is also fluorescent. So these are options for a contrast that we have, and I wanted to show you an example where you can take advantage of a variety of these contrasts. This is a time-lapse movie that was recorded in a publication by Professor Francesco Marangoni here at UCI. It's an image acquired in a tumor of a mouse model, and for visualizing these T cells migrating in the tumor, he's using both intrinsic fluorescence and intrinsic nonlinear signals, such as second harmonic generation signal from collagen, and then the green cells are tumor cells visualized by the two-photon excited fluorescence from NADH and FAD. And these cells, the T cells, are tagged by using a fluorescent protein. So the DNA of the cells in this mouse model was altered to express this fused protein that actually it has two parts, a red fluorescence expressed by all the T cells to be able to track them, but also a green fluorescence part expressed upon calcium binding, indicating calcium activity. This calcium signal helps the cells talk, and it's important to understand the cells' behavior and how they are programmed to interact with different antigens. So that's an example of how researchers use these different contrast mechanisms, like a mix of exogenous and endogenous contrast. So that's okay. I also had this example, which actually emphasizes the importance, the significance of using intrinsic signal that really allows us to image in vivo in human skin. In our case, that's an image that Professor Potma also showed earlier. Is acquired in our lab. So just briefly for people who are not familiar with skin, I wanted to, that's why I have this cartoon here to make sure, since I'm going to talk quite a bit about it, I wanted to make sure you are all familiar with the fact that the skin is structured this way. We have this cellular structure on top, it's called the epidermis, and the fibrillar structure underneath, including collagen and elastin fibers and all other cell populations. The junction between the epidermis and the dermis is undulated to increase the surface of getting oxygen to the epidermis. Only the dermis is vascularized and carrying oxygen. So when we image with two-photon excited fluorescence and the second harmonic generation, we get these types of images. We typically image what we call en face, horizontally, parallel with the skin surface. But there is also an option to image vertically. So that's the way. Let me get here to make sure I use the mouse. So that's the epidermis and the dermis we image. And again, this is the main contrast we use to photon excited fluorescence from keratin and NADPH, FAD to visualize cells. So these are all keratinocytes we capture beneath the skin surface. When we use intensity only, the melanin is much brighter compared to the other fluorophores. So this is what you see in the brighter cells here. And these are the tips of the dermal papilla. That's the name for these protrusions of the dermis into the epidermis. That's that undulation I mentioned earlier. What else? When we go deeper into the dermis, we also have fluorescence from these elastin fibers, and then you can visualize the collagen with the second harmonic generation. And again, Professor Potma talked in detail about the fundamentals of these processes. So again, importantly, that's intrinsic fluorescence, intrinsic nonlinear signals that can allow us to actually use this method in vivo. We cannot stain or label or alter the DNA in our body. So these are the examples for using these different contrasts. Next, I think I wanted to okay. Yes, before I'm summarizing what the fluorescence is good for, I wanted to briefly introduce and justify the need for another contrast mechanism. So the fact that we do this imaging in vivo in clinic allows us to capture all these fluorescence signals, but there is a problem with all these skin components or molecular components we are interested in detecting. Their spectra are very or mostly overlapped, so it's very difficult to. That's an sort of a collection of emission spectra, fluorescence spectra of all these skin components we are interested in, the collagen is separated here. That's the SHG signal of collagen. It's not fluorescence, but it separates very or much easier compared to the other fluorescent signals. You can still we usually work with 2 spectral windows. You can separate a group of these molecular components, but definitely not all of them. So that's generally one limitation of fluorescence microscopy. When you don't use labels, when you use the intrinsic contrast, you don't get much specificity. And one way to enhance this specificity is detecting, resolving in time this fluorescence. So detecting the fluorescence lifetime of the molecule. And again, Professor Potma introduced this a bit, and I also mentioned it earlier. This fluorescence signal, or the molecules in the excited state, I said that once it vibrationally relaxes, here it stays there for some time, and that time frame is in the nanosecond, usually the nanosecond time frame. Well, that time does not have a fixed value. There is some probability involved. The decay is exponential, and the lifetime we measure, this tau 0, is actually an average lifetime. And so again, you've heard it in the lecture this morning, the faster the decay, the shorter the lifetime. Sometimes it's easier to think about it in terms of the decay rate. And so this exponential decay occurs because we have these non-radiative processes that compete with fluorescence. So the decay rate is a summation of this to the radiative and the non-radiative decay. And then we can define a quantum efficiency. That's a measurement for how effective the fluorophore is. And you'll hear more this afternoon from Alex Valmijana in our team about the way we measure this fluorescence and the image analysis we do with the phasor analysis. I just wanted to briefly introduce this concept here. And then just to summarize why fluorescence microscopy is so popular, why do we like to use it in the lab and more recently in clinic. First of all, it adds contrast, right? And this is, I picked these images also relevant to skin imaging, to put side by side the images acquired with multiphoton microscopy, so the types of images I showed earlier with what we get, for instance, from reflectance confocal microscopy. Some of you here must be familiar with reflectance confocal microscopy. The guys coming from WashU, I was thinking. So in reflectance confocal microscopy, you can image beneath the skin surface by collecting the laser light that's scattered from the skin through a pinhole in front of the detector to get used of the out of focus signal to create this optical sectioning. It's a good way to capture the skin morphology. You can get some cellular resolution there, but there is not much contrast. We get these grayscale images. So the analysis and the interpretation is quite limited. So we like the fluorescence microscopy because it gets us contrast. It can also get us specificities through fluorescence microscopy. I don't have a code bar here to show this image here represents the color coded show us different fluorescence lifetime of the pigmented. It would have been good to actually have the side screens, Richard, as well. This monitor had this very poor resolution for my microscopy images here. We cannot turn one of them on. It's fine if you cannot, but anyway, so what we have here, the very dark blue is shorter fluorescence lifetime compared to from melanin compared to fluorescence from keratin on these skin folds. You'll see these images in more detail earlier, but it allows us to detect different components in the skin with more specificity. It can also allow us to do functional imaging like metabolic imaging, and for that I just wanted to show it briefly, the portion of the metabolic imaging that involves cellular respiration. It happens in 3 stages. You have an ADH generated in the first 2 stages, glycolysis and Krebs cycle, that are fed into the electron transport chain and to generate ATP in the oxidative phosphorylation. ATP is the energy that the cells in our body use for different activities. Normal cells have an oxidative phosphorylation dominant type of metabolism. That means more protein bound than ADH, as opposed to tumors that have more glycolysis dominant metabolism, meaning more free NADH, not bound to an enzyme. So this cellular respiration is a very complex process. I oversimplified it here, and I'm bringing up the NADH significance in the process. We are focused on the NADH because we can actually measure the NADH contribution, and through that monitor metabolic changes in cells. We can measure NADH contribution through its fluorescence lifetime. This free NADH that's not bound to an enzyme has a much shorter fluorescence lifetime with respect to the protein bound NADH. And this is what researchers use to look at the metabolic changes in cancer versus normal cells. And this is an example that shows this fluorescence lifetime shifting towards free NADH and shorter fluorescence lifetime in tumors versus normal cells. It's a very useful way. Yes.
And Mihaela, let me interrupt before going into the biological significance. I wanted to perhaps try to connect with a concept that Professor Padma mentioned in his lecture.
Yes, please.
He mentioned that the fluorophores that have quicker lifetime is most likely because there's higher relaxation due to non-radiative transitions. And that often occurs because the fluorophore bumps into other molecules and transfers energy to the other molecules, which allows for a non-radiative relaxation mechanism. And then you just mentioned that free NADH, which is not bound to an enzyme, the fluorescence relaxes faster than when it's bound to an enzyme. So I guess, is it because when it's bound to the enzyme, the lifetime is longer? So
Yes.
is the fact that it's bound to an enzyme basically protect the NADH from bumping into its neighbors and
I think it's
having a non-radiative relaxation?
Yeah, I think it's a bit of both. The NADH, the free NADH are found in more in this glycolysis and more in the cytoplasm of the cells. The protein bound is in the mitochondria, so there is some restriction there. But also the protein bound NADH is a more complex structure.
But it's confined.
But it's yes.
But it is actually, because it's located in the mitochondria, it is more physically confined and in some sense shielded from having a lot kind of random particles run into it and let it release energy. Okay, I just wanted to make that connection. Thank you.
Yes, that's a good observation. So let's see. I said contrast, specificity, functional imaging, and one last important fact is the quantitative analysis, because you can selectively detect, specifically detect these different molecular components. You can also quantify it, and that becomes very important to monitor different changes. And this is an example I selected also from our work, where we showed that in vivo imaging using Flame. Flame is the name of our device that I'm going to talk about next, can capture melanin distribution heterogeneity in human skin. And this is showed in these images. This, for people who are not familiar, this type 1 to 5, this represents skin types. The skin type 1 is the lightest skin. Skin type 5 is the darkest skin. The darker the skin, the more uniform distribution of melanin we have, the more protection we have. And it's very important to capture this melanin distribution and quantify it. We use this approach. So this quantification was done by measuring this melanin volume fraction for these different skin types. We show that we are sensitive to actually being able to distinguish between these different skin types, and then this is a distribution of melanin across the epidermis, and that's important for instance, for applications where we want to monitor the treatment for repigmentation. For instance, we have a study. Go ahead.
So these images on top are en face images?
Yes.
And is it's integrating over the full depth of the epidermis?
I think you are correct. That's actually a projection of over the volumetric, yes, because we acquire, yes.
Thickness, but there is a depth-dependent distribution.
Yes.
Of melanin. And is that what you're showing in B? I'm
and
sorry,
this
I
is,
don't quite
yes,
understand
because
what you're
we
showing in B.
acquired this. Yeah, this is what we are showing here. Sorry, I don't have a point.
So is the x-axis is the percentage of melanin density, and this is the depth?
Yes, this is
so
the
one
depth,
is the surface and 0 is the.
This is here. This is the, yes. This is the depth, and this is the normalized melanin volume fraction. So it shows the increase in melanin towards the basal layer. You have more.
Least at the stratum corneum level, and then it increases as you go towards the epidermal dermal junction.
Yeah, that is correct. And what I was saying is that we need this type of analysis in monitoring the therapy effects, for instance, in therapy for repigmentation. Like we have a study on where we monitor the effects of treatment of vitiligo. Vitiligo is these skin conditions where you have depigmentation in your skin is the skin condition that the famous Michael Jackson had, right? And the way it's treated is with UV therapy. That doesn't work for everyone, and it's important to find out early in the process if it works or not. The therapies are expensive, are time consuming. You wanna know if it works. So that was one example on of quantitative analysis we can do with fluorescence microscopy, and I think that wraps up the first part. Do you have any questions before we move? Go ahead.
This personal question, but like with fluorescence, my understanding is there usually some amount of photo bleaching. So when you take images of the skin, how many images can you take subsequently before photo bleaching starts becoming an issue? For example, for melanin?
Yeah, we don't get into that. We are need to be very careful when we image skin in vivo. We are very careful with the power we use, and also we image fast. And we like to sum up a few frames, so to give time the molecules to relax before you excite them on a second frame, and then we add frames up to increase the signal to noise ratio. But that's a good question, and a concern someone should have when doing this type of imaging in human skin.
Other questions? Yeah.
I guess I'm wondering what the actual imaging setup looks like. Do you need to be like in a...
That's on the next slides.
Okay. I actually have a second question.
Okay.
You mentioned that in the very last slide that you presented with the flame, you mentioned that you were looking at like vitiligo, for example. Have you looked at things like melasma or like other kind of like pigmentation stuff? And yeah, okay.
Yeah, we've also had a study on melasma, but for melasma, we are interested in imaging the macrophages in the dermis. We pretty much go with what the dermatologists are interested in. That's a question they always have when they pick their treatment. Is it topical application? Can they do laser therapy, which involves destructing these macrophages that actually become melanophages,
Right.
because they ingest melanin that's leaking into the dermis. So it's important for them to know whether there is presence of these melanophages in the dermis or not. So this is what we are after during that study. We weren't necessarily quantifying melanin. Here with vitiligo, it's really that the treatment is let's repigment that area.
Yeah. So I'm sorry. So if I understood you correctly, when you're looking at at the applications for melasma with flame, you're looking to see if somebody might develop melasma due to a laser treatment. Did I get that right?
No.
No. Oh.
You're looking what type of treatment you would use. You don't do laser treatment if you don't have a dermal component involved. If the melasma, if the pigmentation is in the epidermis, that's my understanding from the dermatologists I work with.
Yeah. Okay. Yeah. Yeah. Maybe we can talk more about it later.
Yeah. Sure.
Good questions. Other question?
Okay. We can maybe answer questions towards the end. Okay. So a bit about optical and engineering design considerations. And for that, I really wanted to make sure everybody is familiar and understand the very basic setup of a two-photon microscope. Most important thing, the laser source and the beginning and the end are the most important thing, the laser and the objective. We talked about the type of laser that's used in two-photon microscopy, but then you basically have a pair of a pair of mirrors that scans the beam, that scan the beam, and then a system of lenses is called scan and tube lens. These lenses have two roles. One is to image the pivot point of the mirrors on the back aperture of the objective, and also to expand the beam to overfill the back aperture of the objective to take advantage of the full numerical aperture of the lens. This mirror doesn't do much, just steering the beam towards the setup, and then you have dichroic mirrors that separate the excitation from emission, and then this is very simple. Basically, in principle, you can only have one detector, no second dichroic. You can have, I've seen setups with one channel detection, but usually people have multiple channels to be able to separate as much of the fluorescent spectral windows as possible. So that's the standard setup. That's it. It's very simple in principle. When you see these large microscopes on the optical tables, that's mostly because these lasers in the lab, they are tunable. They are commonly Ti sapphire lasers. They have a very large footprint. And then there's a lot of flexibility in terms of excitation and emission. There's also multi-modality detection. So that's the reason that the systems are so large and occupy such a large footprint. But in principle, this is a very basic setup, very basic interaction involved. When we do our two-photon fluorescence microscopy, when we have in mind to do it for clinical use, things become more complex. So on one hand, the setup becomes more complex, but on the other hand, from the engineering point of view, you need to simplify things because you want to have the instrument very compact and portable to be able to take it to clinic. Now, the reason the setup becomes more complex is because we want to optimize different things, and there are many designs out there depending on the application and what exactly you want to use it for. In our case, we wanted to maximize the field of view and speed. And the reason we wanted to maximize the field of view is because in dermatology, most of the lesions you deal with are heterogeneous. And I don't know why. And you need to be able to capture a large area to understand what's going on. A pathologist, when they use their conventional microscope to look at a pathology slide, they use two objectives. They zoom in and out to see what's going on in a sample. So it's the same when we image in vivo. It's very important to have a large field of view. And the reason we introduced this complexity in the system was for maximizing this field of view. I'm not sure everyone is interested here to go through the gritty details of this. I can explain also after the lecture for people who are interested. But basically, we separated the mirrors and introduced many more lenses in there, which we optimized with the idea being of minimizing a motion of the beam at the back aperture of the objective, and also these multiple lenses compensate for the optical aberrations introduced by the large scanning angles that we use for maximizing the field of view. So this is mostly where we made things more complex. And then in terms of the detection is pretty much the same. We have two spectral channels, but also time-resolved fluorescence. Going back to the field of view, so just this setup as shown here maximizes the field of view like the single scanning, right? But you also wanted to, a single scanning with a reasonable objective can get you more than 1 millimeter square max. What we wanted to do was actually scan multiple millimeters square, ideally up to a centimeter if possible. And we are able to do that by using a translation stage that's actually attached to the skin through this customized engineered contraption. Amanda Durkin on my team designed that. So the way it works, we again some from here may be familiar, but the way we use our microscope, we tape a metallic ring to the skin, and that clips magnetically to the imaging head. Hopefully we'll have time to show you a demo this afternoon. We'll show you exactly how it works. Basically, this stage is taking advantage of the skin elasticity, is pulling the skin in the X and Y direction to allow us to image adjacent field of views and then stitch them together. And this is how we manage to increase the scanning area up to 1 centimeter squared. And I'll show you images on that. So just a brief summary of how we go about the design of such a microscope. We first select the objective. That's the most important element of a microscope. And for this clinical instrument, we selected an objective that has low magnification and still have a reasonably high numerical aperture, because we needed to maintain the submicron resolution. Then there are some details related to the beam size that's dictated by the size of the back aperture of the objective, and then you work things backwards. You want to have this is the set of lenses that expands the beam, and you want to keep your magnification to the minimum to be able to compensate to these aberrations. So we needed to work with relatively large beams, but then we also have some limitations from the optics themselves. So then you need to customize the optical design to minimize this optical aberration, to minimize the root mean square of the wavefront error. And what this graph shows here, this is on the x-axis, you have the angle. This is the maximum angle, and it shows that for the optical aberrations are under diffraction limit, except one of them, the defocus, that's related to the field curvature. So that tells us that when we scan large angles, our images have some curvature. You're a bit at a different depth on the side of the image compared to the center of the image. But because we image in thick tissue, that was an optical aberration we can live with, and besides, it can be corrected for if we are interested. And these are the sort of the mechanical translation that we use to do this mosaic stitching. This is more commonly used in the lab when you actually scan the stage with sample on it to get this. It's called mosaic stitching or tiling. This become, when you want to map like a centimeter square, this become pretty slow. So when we image more than, let's say, 3 by 3, I think our threshold was 4 by 4 millimeters squared. If you want to go beyond that, we switch to this mechanism where we use what we call strip mosaic. We park one of the mirrors and we scan with a single mirror and the stage. So we pull the skin under the microscope. On a long distance, we have high-speed stages that can do that. So that's a summary of this design that you need to consider when you put this together. And this is a summary that hopefully that answer your questions about how this system look. This is a system we have now in clinic. It's pretty prototypey. We are working on the next generation of this device, but it's portable and allows us to take it to the bedside. This is a summary of the current performance with a large area scanning that we aimed for, with a single field of view close to 1 millimeter square and the maximum scanning area of 1 centimeter square. We do this imaging rapidly. That's very, very important when you do imaging in clinic. We use a deep learning-based image denoising to recover the noise of the rapidly scanned images. We are in the process of optimizing that. Maintain the submicron resolution, and then we enhance the molecular contrast based on time-resolved fluorescence, as I mentioned earlier. This summarizes our contrast mechanism, the spectral and time-resolved imaging. I'm going to leave Alex explain this to you in the afternoon session. He's also be working with you on a tutorial on this GS Lab, the open source software he developed for analyzing the film images we get. And yeah, this is an example of the types of these volumetric images that we get. Our depth penetration is about, on average, 150 microns, plus minus some tens of microns, depending on the skin type. But generally, we can penetrate down to the superficial dermis. And these are the types of images we get at the macroscopic level, and then microscopic as we get as we zoom in. So we usually get these types of volumetric images, so we have a broad view of what's going on. And these are we currently have 2 instruments developed. One is here in clinic at in our dermatology clinic, and this is what you'll be seeing this afternoon. And another one is in the dermatology clinic nearby in the Gavin Herbert Institute. We are ready to move toward some examples of how what kind of images this microscope take. But let's pause for some questions.
Any engineering questions about the system? Mihaela, I had a question real quickly. Can you go back to the slide where you're comparing the beam expander system with the relay system, and you're talking about the wavefront error? So can you just unpack these two and what maybe the take-home messages are between these two elements?
Yeah, so these are usually again, there are lots of details going on. So usually, remember, we started with two lenses that are generally sufficient to do your job. But once you start scanning these large angles, you introduce optical aberrations. And I'm not sure how familiar people are with, spherical aberrations. You definitely introduce with these types of optics, astigmatism, coma. And this is actually a ZMAX worked out design. And the way it works, it basically allows you to introduce additional optical surfaces and lenses. These are 4 achromats you're using. And you introduce this material one by one, optimizing each of these aberrations. And it's very difficult to do it for all of them.
Maybe you can go back to the overall schematic of your clinical system when you show us where the beam expander and relay system are in the system. So this is this is the beam expander here, and this is the relay?
We have this type of analysis for both of them.
Right.
So
But are you using both elements within as a given system, or only one? You're using both.
Yes,
okay.
Both of them. Yeah.
So one you do to basically maximize the beam so that you fully fill the rear aperture.
Yes, that's a beam expander here.
Right.
And this one here, that's interesting. Again, I didn't want to go into details for the sake of time, and I wasn't sure how interested people are, but I can explain it briefly. So remember, in the previous setup, you have this pair of mirrors that are very close to each other. And then when I say you want to focus the beam on or you want to image the beam of the back aperture of the objective, I said you image the pivot of the mirror. And now the next question is the pivot of which mirror, right? Because they are you have a pair. You usually pick a point in between, and then you have what's happening. You'll have some motion at the back aperture of the objective, and that creates some vignetting. It shrinks a bit your field of view. And what we are doing here, we separate these mirrors. And what this relay system does, it images the pivot of this mirror on this second mirror, and then this expander also images the pivot of this mirror on the back aperture of the objective. Now you have a stationary beam of the back aperture of the objective, and you push on. So this whole system, all it does, it optimizes things. There is no other magic in here. It uses just very basic principle to optimize.
The next slide. So the key issue is that you want, with the wavefront analysis. Yeah. So, I mean, the key issue here is that you want to maximize the angle by which you can have the beam entering the rear aperture so you can maximize your field of view, right? But what it's telling you is that as you have a higher wavefront error, does that mean that at the locations of the image where you're using on the edges of each frame, that you have a slight degradation of your resolution because your wavefront error is slightly higher?
Yes. Okay. Yeah. For this particular aberration. Yes.
Okay. Can you comment on kind of how much you're degrading in resolution?
Yes, we measure that. We
Central versus the edge.
Yes, I think for the 1 millimeter field of view, if I remember correctly, we have, that's in our paper, I think we have 17 micron difference, so close to 20 micron difference. Between the center and the edge, yes.
Okay, yeah. Because I know there are people here that are interested in optical design, so I thought it would be nice to unpack those.
Sure, I'm happy to explain this in more detail after. Just as I said, okay.
Even when you use the beam expander.
Hold on. I'll bring the microphone.
Oh, it was like changing a little bit, but I was wondering if like when you have the like stage that you made for the objective and you're using that like in the clinic, is it like for your objective, is it like a water or oil immersion like objective that you then have to put on like in between the stage when you're putting it on the skin, or like is it just?
Can you? It's an immersion objective. Sorry, I couldn't hear you very well.
Oh,
it's an
sorry.
immersion objective. It's a water objective.
Yeah.
But
So do you have to put that between the stage? then
yes,
for
yeah,
the
I didn't
skin?
go through the details of that,
yeah.
but yeah, that's a good question. We put a drop of water on the skin for refractive index matching, and then the objective is a water objective, but we use an ultrasound gel with a similar refractive index to kind of keep it more in place during imaging. You'll see it in clinic in the afternoon.
Even when we use a beam expander, right, are we still limited by the F number of the objective, or we can go beyond
we overfill the back aperture of the objective, so we use that.
So we're still limited
aperture.
by the objective.
Yes. Yeah. Okay.
Other questions?
Can definitely answer more at the end. Just wanted to make sure I have the time to show you some of the things we can do with our multiphoton microscopy. And again, I picked a limited number of examples for the sake of time, but I'm happy to discuss more for people who are interested. So first, I wanted to show the where this large field of view is most useful, because we can do this macroscopic imaging with microscopic resolution. It's very important for these types of applications in clinic to be able to for studies where we need to monitor treatment. You need to image at different time points. It's very important to be able to go back to the same location. And this is one thing we are interested in for when using this large scanning area microscope. So this is a typical image that we get from the skin. These yellow lines, you don't understand much from this image because it's very zoomed out. It's the yellow lines are the skin folds, so fluorescence from keratin. We have some hairs here. This is just a bubble in our ultrasound gel. It's not a hole in the skin. And then you kind of see these strips. We can correct for them, but many times we don't need it. It's just for the aesthetic. This is so this was acquired on the forearm of a subject. This image was acquired in approximately the same location at a 1 week difference. We don't use any markers, any tattoos. We can just use like a landmark on the skin, a solar spot, or so we just tape this magnetic ring close to that landmark. And then we take some reference point like this hair over here. That's very likely the same hair based on its morphology and the surrounding area. And then they are acquired at slightly different depth, but again, that's not a problem. How do I know it's slightly different depth? Is because we have some blue color showing up here. That's a second harmonic generation from collagen. So you're a bit deeper into the dermis in this one. But then, so once you make sure you are in the same area, you zoom in with our tile mosaic, and you get this type of image. And this is what we've been imaging at different time points to really make sure that we can go back to the same location each week. We show that we can do that with almost cellular resolution, which is quite impressive. That's not to say that this is a cell in here, but it does have the size of a cell. It's a portion of the elastin fiber. So this is very critical and very powerful for these longitudinal studies that we do in our clinical applications. What else? What else can we do? I think this is where I'm going to show some example. One is this approach that Alex really worked out. I'm not sure if he's gonna talk about it this afternoon. He's gonna touch on it, but this is something we've been working out to use this additional molecular contrast that the time-resolved fluorescent brings to distinguish different skin components. So he's using these images. These are images acquired in different from subjects with different skin types, different depths, and the pixels in each of these images carry a lot of information, spectral and fluorescence lifetime. And he uses these high dimension matrices and principal component analysis to reduce this dimensionality and builds up these UMAPs where we can distinguish these different skin components, and that's still some work under development, but we are on the right path with this. And again, he's gonna go into more details about that. Another study, another application we use this microscope for. We have a study that's testing the potential of this approach for early melanoma diagnosis non-invasively. And I like to show this image from a melanoma lesion. That's really visually correct for the field curvature. This is I need to do that. You can really see the tiles in this one. But what's interesting to see is this really captures well the non-uniformity in the image. So when you zoom in, what you are looking at here is a couple of skin folds. This very red color here, these are pigmented cells, and actually melanoma cells and melanocytes. These are non-pigmented keratinocytes. And then we acquire volumetric images of this. I want to go briefly into more details about this. Or am I running out of time? I wanted to give more details about. Okay. About what melanoma is and what exactly we are looking for people who are not familiar with it. So melanoma is a disease of melanocytes in our skin, the cells that carry pigment. And in a normal skin, this is an immunohistochemistry image that specifically stains for these melanocytes. In normal skin, you don't have them that densely packed in the basal layer. You have them more sparse, and besides, they are exclusively located on the basal layer. They are not supposed to be in the epidermis. What's happening in melanoma, when they start proliferating in this basal layer, they also start shooting up. It's called pagetoid spread. And when we image with our microscope, this is what we are looking for, these melanocytes in upper epidermal layers. And when I start this video, I want you to pay attention to start seeing them. And one other thing I wanted to draw attention to is the fact that when you let me see if I can pause this while when it starts. So this is beneath the stratum corneum, the very top surface, and this is where you start seeing these proliferating melanocytes. How do I know they are melanocytes? Because the cells have this these dendrites attached. Remember when I told you we care about maintaining the submicron resolution? That's because we need to image these types of fine structures. We need to be able to identify the melanocytes, and the resolution, spatial resolution, helps us do it. So that's one example, and I think the last example I wanted to show you, I can even point to it in this image here. As we go deeper, you actually start seeing this bunch of cells in the dermis. You don't see them in normal skin, and sometimes in clusters like that. We are very certain that these are some populations of immune cells. Not being very specific, we don't know exactly what they are. We didn't tag them, but we are. This is where our work is focused right now, in trying to understand how we can both detect and distinguish these different immune cell populations. We also started a very interesting study in monitoring the melanoma immune response at the cellular level in patients receiving immunotherapy. And these are very preliminary data, just a representative example that I picked from a patient where we imaged at the baseline before he started immunotherapy. That's again, we acquire large areas, volumetric images. This is just a representative example to show the population of his immune cells in the dermis before the patient started treatment, and then during, at some point during the treatment. And then we do these types of analysis where we look at the number of cells, and then their lifetime. They change in diameter, in shape, and we found some significant differences in these parameters, these 2 different time points. Very, very early to draw any conclusions, but this is something we are working on now. And a few other studies, which I don't have time to go through, you'll hear more about them in the afternoon session. I just wanted to briefly summarize the why is this technology so popular, and particularly for in vivo skin imaging. It's because it offers a really rich source of information, which makes it attractive, because it adds label-free molecular contrast. It allows this non-invasive imaging, the macroscopic and the microscopic with microscopic resolution, if you have the right design. It has the ability to capture metabolic changes at cellular level. It can also capture dynamic. I did not have time to go through that, but that's also a topic we are focused on. And there are always challenges and limitations. In the case of this technology is the penetration depth, and Professor Potma touched on that this morning, explaining how scattering limits the penetration depth, and also we still have to deal with limited specificity, because we do this label free. But these are all, I always like to talk about these challenges, but also opportunities. There are lots of opportunities to work on this to address. Thank you.
It's for Professor Balu.
So you showed some a previous example with I think it was principal component analysis that you used to, I think, decompose the image into the different skin components, right? So the components from the principal component analysis are literally the skin components.
the components from the principal components, and that involves, Alex will be able to explain that involves some guessing, right? In the way we assign those clusters to different skin components.
Yeah, regarding your question, so the components are only two, because the phaser analysis is a bidimensional projection. So we only have two components. That's why the UMAPs have two dimensional. So no, the answer is no. We then have to kind of guess each of those clusters what molecular components they belong to. Yeah.
That's why we work this in parallel, in sort of identifying these different types of clusters, but then also working to identify some of these components we know. That's why we make good guesses, like the keratin or melanin, but then once we get into the world of this immune cell population, there are lots of unknowns in there. So
I mean, the fundamentals of principal component analysis is to identify two orthogonal axes that maximally separate your cell population. And so they're not necessarily directly related to anything physical, but just maximally segregates. It's a classification methodology. Okay, other questions? Okay, well, the good thing is that you're going to see Professor Balu and her group this whole afternoon. So today's kind of afternoon schedule is a little different. We're going to actually walk back onto the main campus, and we'll have two sessions where you get to visit Professor Patma's lab, and they'll do a lab demonstration. And then we have a facility called the Laboratory for Fluorescence Dynamics, which specializes in using fluorescence lifetime imaging and other advanced fluorescence methods to look at various biological systems. So what we'll do is that we're going to just make a short walk over to the main campus. We'll separate this group into two. And so like roughly from 12:15 to 12:45, the two groups will visit one or the other. And then after half an hour, you guys will swap. And then roughly at around 1:30, we'll come back and have our lunch before launching into the afternoon lab sessions. So hopefully you won't be too hungry or be okay to make it 1:30. And so thank you for your attention. And
so they'll have the lunch here, right?
Yeah, we'll have lunch back here starting at around 1:30. That's the goal.
Okay, sounds good.
Thank you, everyone. So, yes. But I want to give you guys maybe 5-10 minutes to use the bathroom or whatever before we just meet out front in the lobby, say at 12:05. Okay.
It's not a dumb question, but when you call the two fluorescence or like the two fluorescent imaging label free, it's the entire point of that modality trying to image these or trying to get that contrast by identifying labels or in like the things that you are imaging. Because I think you were doing like NADH and stuff, and that's like already present in the body. So it's like you're just trying to.
Yeah, but we don't call that label. That's why we call that label free.
Okay, I see. So then in this case, the label is something that you add into the body. Okay.
Yes.
Okay.
Yes. That's interesting. I've never thought about this thing.
No, I think
being confusing.
That's
that's great. That's great. You asked.
Yeah.
So this is what it means. We don't refer to that as a label.
So I mean, I'm wondering if in that fluorescence sense, if you could just like send like almost like a push broom system, use every single wavelength possible, and try to maximize the contrast between two different things that you see in the image, if that makes sense.
You want to excite with multiple wavelengths.